Okay, mechanism of enzyme action. In this you have to learn the theories of enzyme action. There are two theories that is you have to learn for proving this enzyme action. One is ES complex hypothesis. ES complex hypothesis and enzyme substrate interaction. These 2 points. ES complex hypothesis by Michelis Menten theory and enzyme substrate interaction. In that you have to learn 2 hypotheses, that is lock and key and induce with theory. So enzyme substrate interaction, some part was taken in the class, and today we will look with the first interaction method. First enzyme substrate interaction, that is lock and key method. So what is lock and key method? From this diagram you can see that there is a specific shape that you can see in the enzyme, and substrate also modified based upon the shape. So here you can see that the substrate molecule with the cofactor both bind together in order to trigger the activity of enzyme substrate complex to form a product. So we will look at there exists a complementarity and a specificity between substrate and active site of enzyme. So there is a similarity, there is a complementarity, means there is a similarity between the both substrate and enzyme active site. Okay, so there is a complementarity and specificity. So the enzyme shape is rigid, rigid to the what? It is a pre-shaped way the enzyme active site is being made, so that the substrate can easily bind on that what? Predefined enzyme site. Okay, so it can easily bind on that predefined enzyme active site. So this active site of enzyme is fixed, that is the shape is fixed. Okay, there is no any alteration, the shape is fixed. So that because of that fixed one side the substrate will come and bind, and on other side you can see the catalyst also actively bind. So substrate will act as key and active site is the lock. So active site is the lock and substrate is the key. This is the key. So K and L. So another name for this is Fisher's model, and you can see that the substrate is modified, the predefined way the substrate has been modified in order to accumulate our key. Key is the key is our substrate. Okay, so this account for enzyme substrate specificity. So there is the enzyme is predefined in such a way as the substrate can include this substrate molecules. I mean enzyme is predefined so that the it can actively interact with the molecules. Okay, so you can see that this is a enzyme molecule. Okay, don't look this one. Here you can see this is an enzyme molecule, and there is a substrate molecule, and there is a catalyst, and this can actively bind to the predefined shape of the enzyme. So the shape is predefined already, so that only a particular substrate can actively bind to the substrate. So there is an enzyme specificity is there for the particular substrate. Now we move on to the induce with theory that has been proposed by Koshland. So Koshland theory is been asked for the exam. So what is happening when both substrate and active site of enzyme will undergo conformational changes? When the conformational change, until the substrate is completely bound to the enzyme. See, the substrate is completely bind to the enzyme. Here you can see the substrate. Look, this is the enzyme, and the substrate is completely bind to the enzyme. That means there is a change that has been happening. There is a change in the both the substrate and the enzyme is undergoing change, and final shape and even the charge molecule is also varied. So when a substrate and an enzyme come and bind together, there is a conformational change is happening, and this conformational change helps the substrate to bind tightly to the complex bind tightly to the enzyme. So this activity is being initiated by catalytic function. Okay, so there is a catalyst for activating any enzyme. So first point you should know is when an enzyme when the substrate come and bind, there is a conformational change that has been happening. So you can see that there is a precise orientation for functional group in the enzyme. Enzymes are amino acids or proteins, so amine group can be seen, isn't it? So this amine group, functional group can be what? It has been changed. It has been there is an orientation, there is an arrangement of functional group which is also necessary for enzyme substrate complex formation. So there is a reorientation and conformational changes that has been happening when there is a attachment of substrate to the enzyme. Okay, so when there is a attachment of substrate to the enzyme, there is a conformational change that is happening and also reorientation of the functional group of enzyme. So an enzyme substrate formation result in a strain. Because of the strain only conformational changes is happening. So when enzyme substrate complex form, it is transformed to EP. What is EP? Product. So this, since the strain is there, since a conformation change is happening, this is also called as substrate strain theory. Okay, now we are moving to the factors affecting the enzyme reaction. First you have to learn is the temperature. Temperature is the common factor that affect the enzyme reaction. If there is an optimum temperature, only enzyme catalytic reactions take place. Okay, so which is an optimum temperature condition? For animal enzyme it is 20 to 45 degree for good activity. For good enzyme reaction, 20 to 45 degrees ideal. For plant enzyme, 40 to 60 degrees very good. Otherwise, when the temperature increases, the protein can undergo denaturation and it can destroy also. Sometimes temperature, lower temperatures, negative temperatures can also cause a destruction. This is the temperature is increasing the rate of reaction at a good temperature, that is 35 to 40 degree for human enzymes. 35 to 40 degree, that is an optimum temperature where the enzyme will show maximum activity. See, this is the optimum temperature where the enzyme showed maximum activity. So in lesser than 5 degree it is inactive. Okay, and it is above 40 degree, in some enzymes it causes denaturation, that is destruction. So what is the effect of after temperature? We are just moving with pH. What about the pH? Here again an optimum pH should be there. For different enzymes there is an optimum pH. So if the for cellular enzymes, if that is close to neutral value, it is very active. For high temperature or low pH, it can slow down the reaction, and it can very high temperature can also cause destruction of enzymes. So strong acid bases can also inactivate enzyme permanently. So this is an ideal. Neutral pH is considered to be an ideal pH, where the physiological pH where the enzyme substrate complex formation is ideal. Okay, now next we are moving to effect of substrate concentration. And here comes the role of Michelis-Menten. Effect of substrate concentration is affecting the reaction. So before that you should know what is Michelis-Menten constant. So according to Michelis-Menten, enzyme reversibly combine with substrate to form. So enzyme substrate complex is formed. So E plus S give ES. Okay, and ES can be dissociated into E plus S. Okay, so there is a reversible action that has been taking place, which can yield a product by releasing free enzyme. Okay, it can yield a product by releasing the a free enzyme. In your textbook it is given K1, here K2, and here K3 is given. Okay, so what is S? S is the substrate, E is the enzyme, ES means what? Enzyme substrate complex, P means product. And what about K1? K1 is the forward reaction, and K2 is the, here 2 only, K2 is the reversible reaction, and K3 is the product formation reaction. All these are rate constants. Okay, rate constants. So when we talk about rate constants, how speed up the reaction is taking place. So rate of the enzyme reaction is directly proportional to substrate concentration. That you should keep it in your mind. It is always first order reaction only. It depends upon the it doesn't depends upon the concentration. Okay, so it is a first order reaction. So Michelis-Menten equation is always what has been plotted in a diagrammatic representation. Okay, so there is a Michelis-Menten plot, and this is the equation: V is equal to Vmax into substrate concentration divided by Km plus substrate concentration. So that we will deal now. So you should understand that what the substrate concentration, the rate of reaction of enzyme reaction is directly proportional to substrate concentration. See, there is a rate of reaction increases. It is directly proportional to. So rate of reaction directly increases with the substrate concentration initial velocity time. Initial velocity means what? V0, starting velocity. For any enzyme reaction to happen, there should be a initial velocity. So you can see that in some point it has reached a maximum value. So the velocity of reaction that reaches the maximum velocity is called Vmax. So in Vmax condition, what happens? There is a enzyme substrate that has been formed in a maximum velocity stage. Okay, maximum velocity stage. So at this stage, the velocity of reaction is independent of substrate, but it is proportional to enzyme concentration. So you can see that there is substrate, there is enzyme. So enzyme substrates are always been in complex formation, but there is a point where saturation occurs. Now no more substrate cannot interact. Okay, so that saturation point is happening. That is the what we call it as Vmax value. So there will be, whenever we add more substrate, there will be no effect on the velocity. Why? Because there is no any formation of Vmax, or there is no any formation of ES, or that is zero order kinetics. So whenever there is an increase in substrate concentration, here you can see more substrate are seen, but enzymes is almost in saturation point. So here in that condition there will be no effect on the velocity. So that is why it's showing some sort of steadiness. Okay, so we were talking about the Michelis-Menten equation. Okay, so according to this equation, we can say that velocity varies with substrate concentration. Okay, velocity can change with substrate concentration. So Michelis-Menten constant, the substrate concentration of the velocity of a reaction is half of its potential maximum. It is called as Michelis-Menten constant, where K stands for constant and M stands for what? Michelis. Okay, so a maximum velocity is reached. So another name for that is it is always having a first order kinetics and also a zero order kinetics when it reaches the halfway. See, here you can see zero order kinetics, and starting initial stage you can see the first order kinetics. So here we can say that it is depending upon the concentration, but initially they are not depending upon the concentration. Since initially they are not depending upon concentration, we are calling it as a first order kinetics. But if it is when we move on to further process, enzyme substrate complex is forming, enzyme and product is being dissociated. So in that condition you can say that it is having again a second order reaction, and last zero order reactions can also be observed. Okay, so from you get an idea how the enzymes and substrates are interacting with each other. Okay, and the formula for this is that you should learn: V0 is equal to initial velocity, V0 is equal to Vmax into substrate concentration. Vmax means maximum velocity where substrate and enzyme form a complex, a maximum velocity required for substrate enzyme complex formation. Okay, so this is understanding: Vmax into substrate concentration divided by Km plus substrate concentration. This is an equation for calculating the overall velocity of enzyme reaction. What is V0? V0 is the initial velocity. So there is a point where you can see that See, look at this. There is a point where you can see that there is a half Vmax value that is in. Okay, so first of all, an initial velocity has been happening. Initial velocity, enzyme substrates are equally interacting with each other, and in maximum velocity, it is in saturation point. But in between them, you can see a half Vmax value or Km value. We are saying as Km value. Half the number of enzyme molecules will be in the form of enzyme, and others will be in the form of ES. So what is half in this time? What is happening to enzyme and substrate? This half Vmax time or Km value. During Km value, what is happening? Half of the enzyme molecule will be in E form and others will be in ES form. Okay, because from down you are getting substrate now, so you will have half of the enzyme will be as such. Okay, as such, and half of the there will be complex formation. Okay, that is half Vmax value. This is half the maximum velocity. So half Vmax value, another term is Km value. K is the rate constant and M is the Michelis constant. Okay, Michelis Menten form. So this is V0. Okay, V0. So one more time I will say, yeah, enzymes are there and the substrate are there, both are interacting together. Initially they show only a first order reaction because it is concentration independent, and when they reach halfway round, they reaches half Vmax value or Km value, where the enzymes are as such. Half the number of enzymes are existing as E, and half the number of enzymes is always seen in enzyme substrate complex or EP formation or in enzyme product form. Okay, because we have started our work, we have started enzyme and substrate together. Okay, so that is why we have to know that Km value is different for different enzyme. Okay, Km value is different for different enzyme. So substrate concentration need to be saturated with enzymes. So Km value is very important. Km value is also called as half Vmax value. It is inversely proportional to affinity of enzymes for substrate. So lower the Km value, higher will be the affinity. So higher the Km value, lower will be the affinity for enzyme substrate complex formation. So this is the factors affecting this reaction, enzyme substrate complex reaction. Hope it is clear. And this class we have deal basically on enzyme substrate interaction.